Well, let's go ahead and get underway. I want to extend a welcome to those of you who have joined this morning. This is, as the slide indicates, and hopefully you can all see my slides that I'm showing. If you can't, please let me know. But this is, of course, the session on blood vessel damage and injury, as the slide indicates. And so I'm going to talk a little bit about what the lab that I direct here at the University of Utah studies, and hopefully be able to do that clearly and concisely, and then be able to open it up for any questions that any of you may have. So I'm going to go ahead and start with something that we that we see all too often. I like to watch football myself, and this is something that happens more often than it should. Fortunately, I think with some rules changes and such, these incidents are becoming fewer and fewer, but it's still a rough game. Of course, this player suffered a traumatic brain injury, so I want to talk a little bit about that. And actually, the only reason I know that he suffered a traumatic brain injury is that he, excuse me, appears to have lost consciousness, right? So when people suffer a traumatic brain injury, that's something that commonly happens. That is a loss of consciousness. That's a manifestation of a neural injury, right? The brain is made up of nerves, and if those nerves get damaged or temporarily injured, then they stop functioning the way that they should, and the loss of consciousness is a manifestation of that. So this next image, I'm just going to warn you in advance, is going to be a picture of a human brain, okay? And it's an actual brain, but it's a nice clean image. I don't think it will be offensive, but if you're a little squeamish or something, then just be prepared. All right, so this is a cadaver brain, a brain that somebody once used but no longer needed. And as we talk about what happens to a brain in traumatic brain injury, of course, the neurons can be injured, right? The other thing that can happen, as you can see on this brain, there are a number of blood vessels that are also coursing around the brain, right? And these vessels, when the brain gets injured, these vessels can also be injured, okay? And they're not just on the outside of the brain. If we were to dissolve away the surrounding brain tissue, then we would just be left with the blood vessels. And you can see that there are many, many blood vessels, both around and throughout the brain. So it becomes a matter of potential significance with injury. When someone does suffer traumatic brain injury that includes vascular injury, there are a few things to worry about. The first of those is structural disruption of vasculature. So, and right now, let's talk initially about things that you need to worry about right away. I'm going to call those acute challenges, all right? So, structural disruption is a common structural disruption of blood vessels in particular is a common consequence of traumatic brain injury, and it can manifest itself as contusion. Okay, contusion is just another word for bruise, but it's commonly used to describe bruises on the surface of the brain where the brain has interacted with the inside of the skull as a result of a traumatic event, okay, a mechanical event that pushed the brain up against the skull and caused injury. And of course, I'm showing here a CT scan, something that physicians would do with a patient in the hospital if they're trying to figure out whether they're injured and how severely they're injured. A CT scan can also reveal intracranial hemorrhage, which is just overt bleeding inside the head because a blood vessel has been disrupted. Okay. The other thing that can happen is blood vessels, as you may or may not know, have smooth muscle cells in their walls. And those smooth muscle cells make it possible for a blood vessel to contract and to dilate to control the flow of blood flow, okay? So, but the other thing that is observed acutely after many traumatic brain injuries is dysfunction of this ability of the blood vessels to control blood flow inside the brain. So usually the brain is very good at controlling blood flow, but not so good after injury in many cases. So it's clear that hemorrhage is a consequence of vessel damage, right? The bleeding occurs because the blood vessel walls got torn and they're now leaking. But we could ask the question, what if the vessel isn't bleeding? Does that mean everything is okay? Well, dysfunction, as I mentioned on this previous page, is often observed in cases where there is no obvious hemorrhage. So it seems that the blood vessels can actually be injured and become dysfunctional even if they're not bleeding. Okay, and so that's another issue that we need to try to think about and understand. Now, traumatic brain injury also has a lot of consequences that are longer term problems. Okay, people who have suffered traumatic brain injury are known to have ongoing headaches, to suffer from second impact syndrome, which means they're more susceptible to a subsequent injury, and an increased risk of stroke, etc. Lots of potential problems. Okay, so one of the questions that I and my lab that we're very interested in is, could vessels that are damaged, maybe subtly, right, so they're not bleeding in a traumatic brain injury event, could they contribute to some of these long-term challenges? All right, so the goal of our laboratory is to define what happens to vessels during trauma. And if we ask ourselves, how do we hope that this will change the world? There are a couple of things that we think, a couple of ways that we think we can contribute. One is to provide insights that may be needed for vascular therapy after a traumatic brain injury event. Currently, there's nothing done to treat blood vessels that aren't bleeding. Okay, as long as blood's still moving through them, then we just hope the patient recovers. The other thing that we think this research will do is to improve injury prediction and definitions of injury tolerance. So by that, I mean provide information about how we can better design helmets and automotive interiors and that sort of thing to better protect people in known dangerous situations. So these hopes lead us to two fundamental questions. One is, what do vessels experience during trauma, and how do they respond to this loading? Okay, so I'm just quickly going to show this image of a blood vessel so you can appreciate a little bit of its structure if you don't already. There are three distinct layers in almost all blood vessels: inner, middle, and outer, intima, media, and adventitia. Okay, and these layers have structural features to them, kind of like a woven fabric, if you will, made up of proteins that provide the vessel its mechanical strength. And there are also cells that are intermingled with these layers to provide function to the blood vessel. Again, for example, to allow it to contract and dilate. Now, when we test, or in order for us to answer many of these questions, we actually do a lot of testing, a lot of experiments on cerebral blood vessels that have been dissected from a body. Okay, whether those come from humans or from animals, whatever it may be, we isolate them from the body, and then we, as you can see in this image here, we tie the vessel to needles at each end. And once we've done that, we can pressurize the vessel and we can stretch the vessel. Okay, and we can measure the pressure and we can measure the force that's required to stretch it. We can also measure the amount of deformation or shape change that the vessel undergoes with the applied loading, right? The pressure and the stretch that we apply. This is an example of such a test. This is a cerebral artery from, I think, from a pig brain, actually. But you can see that we're testing it all the way to failure, and so it tears in the center. The size of this vessel, just to give you some perspective, its outer diameter, or its width, if you want to call it that, is about 1/2 millimeter. Okay, so this is we're really zoomed in with a microscope to be able to visualize this. Okay, so one thing that we can do with these tests is define the mechanical properties of a blood vessel, or in other words, determine how the force that the vessel experiences as it is stretched changes. Okay, and so this is a plot that shows that, well, it shows stress versus stretch, but you can think of that as the force that a vessel is experiencing as it's being stretched. Okay, now these mechanical properties can be utilized to help build computational models of the brain so that we can try to predict whether or not an injury would occur under certain conditions. And so along with some geometry of blood vessels, for example, this is actually the geometry, representation of the geometry of the blood vessels in a rat brain, but incorporating geometry and material properties into a computer model then allows us to predict the level of deformation and potential injury there is in a brain under a given amount of loading that we're studying. In this first row here, you see the strain or the deformation that's associated with the brain during blast loading or from an explosion as a function of time. So this is taken at different times. You can see from 0.8 milliseconds to 3 milliseconds, right? And on this second row, you can see the results of deformations in the blood vessels. So that's one way that our research is utilized by many to help us better understand traumatic brain injury. But we also, as I mentioned, use this model of overstretch in the lab to try to understand how a vessel being stretched may change its performance, okay? And as I mentioned before, a blood vessel has not just structural characteristics, and therefore we need to define its mechanical properties, but it has functional characteristics. Again, this contracting and expanding, relaxing, okay, of the smooth muscle. So some things that my group has done to address these questions include one that was reported in this paper a few years ago. The primary investigator here was Dave Bell, a student who's graduated from my lab with a PhD and now is working at BD, actually. What he did was attach a blood vessel to needles and then expose it to a series of concentrations of a chemical, in this case potassium chloride, and potassium chloride stimulates the smooth muscle cells in the vessel wall to contract. So you can see this healthy blood vessel here, as it experiences greater concentrations of KCl, it contracts more and more, right? So what he did is he took a healthy blood vessel, actually this was from a rat, but a healthy blood vessel, a number of healthy blood vessels, and studied their response under the healthy state, and then stretched that blood vessel by some amount, and then re-performed the experiments with the exposures to the chemical. Okay, and what he observed is that there were some changes in the amount of contraction that the blood vessel experienced as a function of the concentration of the drug. Don't worry about the left side of this slide. This is just a measure of how we can quantify that percent contraction. But what he found was that there really wasn't a change in the behavior of the blood vessels of its smooth muscle cells when the vessel was exposed to small deformations, whether they were slow or fast. But he began to see significant changes when that vessel was stretched to a further extent, and especially at higher rates. Okay, so that was an interesting thing for us to observe, that the blood vessel became functionally, it was altered more, right? The function of the blood vessel was altered more when it was stretched to by more extreme amounts and at higher rates. So that, of course, suggests that maybe that sort of change could be expected in a traumatic brain injury. A question that this same student, Dave Bell, also pursued was to ask whether or not the mechanical properties of a blood vessel would change after a stretch. Okay, so he performed a very similar experiment where he took blood vessels and, again, studied their mechanical properties under their healthy state. So this again is showing, this is stress versus stretch, but again, you can think of this as the force that the blood vessel is experiencing as it gets stretched. And then he overstretched the blood vessel by some amount and then retested the behavior of the blood vessel. And you can see that its performance after the overstretch is different than its performance before. And in fact, if one way that you could think about this is sort of like what happens to a balloon. If you take a new balloon out of a package, maybe it's a small balloon, right? So it hasn't been inflated yet, and the balloon is little, and then you blow it up, and if you let the air back out of it, then that balloon is now all stretched out. And in fact, that is essentially what we see happening with these blood vessels after they're stretched out. And so again, you can imagine if a blood vessel experiences that kind of overstretch during a head trauma event, then the way that it performs in the body, the way that it performs its function in the brain thereafter, we would expect to change. Okay. All right, so I'm just going to skip through this next slide because I think I've given you the essence of those experiments, but it's worth asking, okay, what is at the root of the observed changes? Maybe if we can figure out how this overstretch has changed the blood vessel, it will give us some insight into how we can maybe treat the blood vessel to help it recover and perform its proper function. And this is a question that we continue to work on trying to answer. And we don't have all the answers yet, but we do have some insights that we can report. And this work was primarily done by Matt Converse, reported in this paper shown here. But Michael Yu, who is a professor in bioengineering, invented a synthetic molecule, which he calls collagen hybridizing peptide. And he calls it that because it has the same molecular structure as a collagen fiber does. Okay, so if you look at the different levels of structure in a collagen fiber, at the molecular level, we see that it's made up of these three helically shaped molecules that wrap around each other. Okay, so he built this collagen hybridizing peptide, this synthetic molecule, to have the exact same molecular structure, such that if a collagen molecule is disrupted at all, then if that molecule is exposed to these collagen hybridizing peptide molecules, they'll actually bind to one another because their shape is complementary. Okay. Dr. Yu also recognized that he could attach a fluorophore to this molecule, and then by virtue of that fluorophore, be able to identify where his synthetic peptide had attached to the collagen molecule, which was the damaged collagen molecule. So my student, Matt Converse, wondered if this could be used to help us identify collagen fibers that may be damaged in the wall of the blood vessel after it's overstretched. So he did an overstretch experiment and then incubated the vessel wall within this collagen hybridizing peptide. Okay. And sure enough, showed that indeed the collagen fibers of the vessel wall are damaged as a result of this overstretch. And using this molecule, we can identify where that damage occurs. Okay, so Matt studied experiments or performed experiments on blood vessels with both axial overstretch, that's what this bottom row is, so stretching a vessel along its length, and circumferential overstretch, where a blood vessel is expanded circumferentially. Okay. And you can see, looking left to right here, that the images get brighter and brighter, and you can see more evidence of collagen disruption by virtue of this fluorophore attached to the collagen hybridizing peptide. So this tool, and you can see the same thing in the axial overstretch experiments, though the nature of what we can see at these two due to these two stretch directions is a little different, just because the the nature of the collagen fibers is a little different in these two directions in the blood vessel. But this tool is super powerful because now it allows us to not only identify that collagen is being damaged, but we can identify how much collagen is being damaged and where, okay? So this is a really busy slide, but I just want to point out quickly here then that, so these black dots are all they represent the amount of damage that Matt measured in a given blood vessel that had been exposed to a certain amount of stretch. And he was able to see that blood vessels can stretch a certain distance before collagen starts to be damaged. But then once you pass this threshold, the tendency for the collagen to be damaged increases dramatically with additional stretch, okay? And he also observed that if we plot these points on top of the mechanical behavior of the blood vessel or its mechanical properties, you can see that the vessel starts to fail, or its properties start to weaken at this same threshold where the collagen damage was identified. So that's a pretty interesting finding, we think, that we're trying to learn more about, and that again will hopefully lead us to some insights about how vessels that have been damaged can be treated, okay? All right, so one other thing that I want to mention here is, so I've talked all about traumatic brain injury so far, and of course vessel injury in the context of traumatic brain injury. But another area that we recognize our research may be useful is that of balloon angioplasty. And you can see I took this image from vector stock here just to demonstrate the idea of balloon angioplasty, but physicians use balloon angioplasty when a blood vessel gets diseased, right? So this is the kind of disease that develops in coronary arteries. And as this plaque that's developing in the vessel grows, eventually it cuts off blood flow through the artery. And when that happens in a coronary artery, then the heart is no longer getting blood and you have a heart attack, okay? So in order to treat this, one thing that, well, and I should say this disease phenomena occurs throughout the body, not just in the heart, including in some of the blood vessels of the brain. But when this occurs, physicians will often take a catheter and get it, and they'll introduce it into your circulatory system at, for example, your femoral artery in your leg, and then they're able to direct it up into the heart or into your brain or whatever they need to do. And they direct it to the point of the disease, and then there's this balloon that they can blow up from back at the attachment to their catheter and force the blood vessel open, force this plaque open so that blood will start flowing more freely through that region. And then oftentimes there will be an expandable stent, a little metal cage sort of, that gets expanded in the blood vessel that they leave there to try to keep it open. But this process invariably causes damage to the blood vessel wall. And there's actually very little known about what the optimal parameters, for example, of this procedure should be. So for example, physicians don't have much scientific guidance on, what the ideal amount of stretch is for this blood vessel to experience. Clearly, if they overstretch, they run the risk of rupturing the blood vessel. If they understretch, then they run the risk of not accomplishing anything at all. So there's a lot that still needs to be learned about this procedure, and we're starting to study this in our lab. And this is just a little video of an experiment in the lab where we expanded a blood vessel using a balloon, such as might occur in angioplasty. This is actually a pig carotid artery. And you can see that as that balloon continues to expand, it eventually just ruptures the blood vessel wall, right? Now, of course, that would be a very bad outcome of a medical procedure, and physicians are careful not to let that happen. But again, we're doing research into that to hopefully provide a little more guidance. Okay, so in conclusion, I just want to acknowledge, I mostly sit at my desk in my office, and write stuff, write proposals to try to get government agencies to give us money so we can do research. I want to acknowledge the students who make up the lab. I've been lucky and blessed to work with lots of great students during my time at the University of Utah. I'm highlighting those whose work I highlighted today. I also want to acknowledge my collaborators in this, and also funding sources. And I'd be happy to take any questions if any of you would like to ask questions.